Close your eyes and imagine the most beautiful book ever written. Picture pages filled with elegant script, each letter carefully placed to tell a story of wonder. Now imagine that this book lives inside you. Not tucked away on a shelf, but woven into every fiber of your being. This book exists. It's written in a language older than any human tongue. More precise than any computer code. More personal than your own signature. The language has only 4 letters. A. T. G. C, that's all. Just 4 simple characters, like the building blocks of a child's alphabet game. Yet with these 4 letters, nature has written the most complex and beautiful stories in the universe. Right now, as you rest here in the quiet darkness, trillions of these letters are dancing through your body. They spiral through your cells like microscopic tornadoes, carrying messages that keep your heart beating. Your lungs are breathing. Your mind is dreaming. A stands for adenine. T for thymine. G for guanine. C for cytosine. These names might sound strange to your ears, but they are the most familiar words your body knows. More familiar than your own name. Your cells have been reading these letters since before you took your first breath. Think of your favorite poem. The way certain words flow together, creating rhythm and meaning. The genetic alphabet works the same way, but instead of creating verses about love or loss, it creates you. Every freckle on your skin. Every curve of your smile. Every unique thought that crosses your mind. The letters combine in groups of 3, like tiny haikus written in molecular ink. ATG might tell your cells to start building a protein. TAA whispers that it's time to stop. Each 3-letter word has its own meaning, its own purpose in the great symphony of your existence. Scientists call this the genetic code, but that name feels too cold for something so warm and alive. It's more like a love letter that life wrote to itself. A song that began 4 billion years ago and still plays in every living thing on Earth. Your genetic alphabet connects you to everything that has ever lived. The same 4 letters that spell out your existence also wrote the story of the mighty oak outside your window. The family cat curled up nearby. The flowers that bloom each spring. When you breathe in, you're sharing air with countless other beings, all of them carrying the same 4-letter language in their cells. It's as if all of life speaks the same secret tongue, a molecular conversation that has been going on since the first cell learned to copy itself. But your personal copy of this language is unique. Like fingerprints or snowflakes, no 2 genetic stories are exactly alike. The 4 letters arrange themselves in your cells in a pattern that has never existed before and will never exist again. You are a limited edition in the library of life. The total length of your genetic story is staggering. If you could unwind all the DNA from just one of your cells and stretch it out like a ribbon, it would reach about 6 feet long. Now imagine that this 6-foot ribbon is packed into a space smaller than the period at the end of this sentence. It's like fitting the entire works of Shakespeare into a grain of sand. But the true magic isn't in the size of the story. It's in how it gets told. Every cell in your body contains the complete tale, all 6 billion letters of it. Yet each cell only reads the chapters it needs. A heart cell reads the parts about pumping blood. A brain cell focuses on the sections about thinking and dreaming. A skin cell studies the passages about protection and healing. It's like having a vast library where every book contains the same story, but each reader sees different meanings depending on where they are and what they need to know. The story remains the same, but its expression changes like light through a prism, creating a rainbow of possibilities. Your cells read this genetic language constantly, translating its molecular words into the proteins that build and rebuild you every moment. As you sleep, teams of cellular readers are working through the night shift, following genetic instructions to repair the day's small damages and prepare for tomorrow's adventures. The language flows through you like a river of information. It carries memories from your ancestors, instructions for your present, possibilities for your future. It connects the person you were as a tiny embryo to the person you are now, and to the person you're still becoming. Tonight, as you drift toward sleep, you carry within you the most intimate story ever told. Written in letters of life itself, it speaks of your connection to every living thing. It whispers of the incredible journey that brought you to this moment, and the amazing truth that you are, quite literally, poetry in motion. But this ancient language holds more secrets than we've yet discovered. Deep within your cells, there are chapters still being written, stories still unfolding, and mysteries that science is only beginning to understand. Imagine stepping into the oldest library in the world. Not the ancient library of Alexandria or the weathered halls of Oxford, but something far more remarkable. A library so small you could never see it with your naked eye, yet so vast it contains the wisdom of 4 billion years. This library exists inside you right now. In fact, you carry trillions of copies of it, one tucked away in nearly every cell of your body. Each copy sits in a special chamber called the nucleus, protected like precious manuscripts behind cellular walls. The books in this library don't have leather covers or paper pages. Instead, they're written on ribbons of DNA that twist and coil like ancient scrolls. These molecular scrolls contain the most important collection of instructions ever assembled. Not recipes for bread or blueprints for buildings, but the plans for creating life itself. Each book in your cellular library tells the story of how to build something essential. One book might contain the instructions for making your eyes. Another holds the secrets of your immune system. A third describes how to construct the proteins that carry oxygen through your blood. But here's what makes this library truly extraordinary. Every single copy is identical. The book about building hearts sits in your toe cells, even though your toes will never pump blood. The instructions for growing hair rest quietly in your liver cells, patiently waiting, never to be opened there. It's as if every room in a vast mansion contained the complete plans for the entire house. The kitchen holds blueprints for the bedroom. The attic stores designs for the basement. Each space carries the whole, even though it only uses the parts it needs. Your genetic library didn't appear overnight. It's been growing and evolving for longer than you can imagine. The first tiny collection began almost 4 billion years ago, when the earliest cells learned to store information in DNA. Since then, the library has been carefully maintained, copied, and passed down through an unbroken chain of life. Think of it like the world's longest game of telephone, but one where the message stays perfectly clear. Your parents received complete copies of their genetic libraries from their parents. Before passing them to you, they created new combinations, mixing their collections like a master librarian organizing the perfect anthology. Every time a cell in your body divides, it faces an enormous task. It must create a perfect copy of its entire library before splitting in two. Imagine a monk in a monastery, carefully copying every letter of every manuscript by candlelight. Now imagine doing this for 6 billion letters, with nearly perfect accuracy, in just a few hours. Your cells perform this miracle of copying constantly. As you sleep, teams of molecular scribes work through the night, reading each genetic letter and carefully placing matching letters alongside them. A matches with T, G pairs with C. Like a dance where every step is perfectly choreographed, the letters find their partners and create identical copies. But this ancient library holds more than just instruction manuals. Hidden between the practical guides are love letters from your ancestors. Genetic poems that describe the color of your grandmother's eyes. Molecular songs that carry the rhythm of your great-grandfather's heartbeat. Stories of survival that stretch back through countless generations. Some sections of your library contain chapters that haven't been read in thousands of years. Ancient instructions for growing thicker hair in cold climates. Old recipes for processing foods that no longer exist. These genetic fossils sit quietly on your cellular shelves, reminders of the long journey that brought you here. Other parts of your library are brand new, written just for you. Unique combinations that have never existed before. Fresh chapters that tell the story of your particular way of thinking. Your specific way of laughing. The exact shade of brown in your eyes or the precise curve of your smile. The librarians who maintain your genetic collection work with incredible precision. They patrol the molecular shelves, looking for any damage or errors. When they find a problem, they carefully repair it, ensuring that the precious information stays intact. Sometimes they make tiny improvements, adding new notes in the margins or updating old instructions. Your cellular library isn't isolated. It's part of a vast network that connects every living thing on Earth. The books in your collection share chapters with the libraries carried by oak trees and dolphins. The same fundamental stories appear in the genetic collections of butterflies and blue whales. This shared heritage means that you're reading from the same ancient texts that have guided life for billions of years. The instruction for making proteins is nearly identical whether it's found in your cells or in the cells of a mushroom growing in a distant forest. You're all part of the same vast library system, checking out different books but drawing from the same cosmic collection. As the night grows deeper and your breathing slows, consider the quiet miracle happening inside you. Millions of tiny libraries, each containing the complete story of how to build and maintain a human being. Each one a treasure trove of wisdom accumulated over countless generations. The books are being read right now, their instructions carefully followed by cellular readers who translate genetic text into the living poetry of you. But who are these dedicated translators, and how do they transform molecular words into the proteins that dance through your dreams? Picture a peaceful morning in a small village where everyone speaks different languages. French bakers, Italian musicians, Japanese gardeners, all living together in harmony. In the center of town sits a wise translator who helps everyone understand each other, turning words into actions and dreams into reality. Your cells are like that village, and deep within each one lives the most talented translator in the universe. This molecular interpreter has one extraordinary job. It reads the ancient language of DNA and translates it into the busy, practical language of proteins. The translator's name is RNA, and it works with the patience of a master craftsperson. Unlike DNA, which stays safely tucked away in the nuclear library, RNA ventures out into the world. It carries genetic messages from the protected inner chambers to the bustling factory floors of your cells. Think of RNA as a gentle messenger walking between two worlds. In the quiet library of the nucleus, it carefully reads genetic sentences, letter by letter. Then it travels to the protein-making workshops in your cell's cytoplasm, where it shares what it has learned. But RNA doesn't just copy the DNA's message word for word. Instead, it performs an elegant translation. Where DNA uses the letter T, RNA writes U instead. It's like changing from one alphabet to another, maintaining the meaning while adapting to a new form. The process begins when your cell decides it needs a particular protein. Maybe your muscles need more of the proteins that help them contract. Or perhaps your immune system requires proteins to fight off a cold. The cell opens the right book in its genetic library and begins to read. A molecular machine called RNA polymerase approaches the DNA like a careful scholar approaching an ancient text. It unwinds the double helix gently, separating the two strands like opening a book to the right page. Then it begins the delicate work of copying. As RNA polymerase moves along the DNA, it reads each genetic letter and places the matching RNA letter alongside it. A pairs with U. T pairs with A. G pairs with C. C pairs with G. The process unfolds like a slow dance, each step perfectly choreographed by millions of years of evolution. The newly created RNA message is called messenger RNA, or mRNA for short. Like a handwritten letter carrying important news, this mRNA contains the exact instructions needed to build a specific protein. But the message is still written in the language of genetics, using three-letter words that need further translation. The mRNA leaves the nucleus and travels to a remarkable structure called a ribosome. If the nucleus is a library, then ribosomes are the workshops where genetic blueprints become reality. These tiny factories float through your cells like wise craftspeople, ready to build whatever proteins your body needs. The ribosome reads the mRNA message 3 letters at a time. Each 3-letter word, called a codon, is like a piece of a molecular puzzle. August means start building here. UUU calls for a building block called phenylalanine. UAA whispers a stop, the protein is complete. But how does the ribosome know which building blocks to use? This is where another type of RNA enters the story. Transfer RNA, or tRNA, acts like a delivery service for protein parts. Each tRNA molecule carries a specific amino acid, the basic building blocks that link together to form proteins. Picture tiny boats floating through your cellular waters, each one carrying a different cargo. One boat brings glycine, smooth and simple. Another carries tryptophan, large and complex. A third delivers serine, ready to help proteins fold into perfect shapes. Each boat knows exactly which genetic word calls for its particular cargo. As the ribosome reads the mRNA message, the right tRNA boats dock at exactly the right moments. The amino acids they carry link together like beads on a string, forming chains that will eventually become complete proteins. The process happens with incredible precision, rarely making mistakes despite its complexity. The growing protein chain doesn't stay straight like a string of beads. Instead, it begins to fold and twist, guided by the chemical properties of each amino acid. Some parts are attracted to water and reach outward. Others shy away from water and curl inward. The protein finds its perfect three-dimensional shape like origami folding itself. This folded protein might become an enzyme that helps digest your dinner, or a hormone that carries messages between different parts of your body. Perhaps it will join the scaffolding that gives your cells their shape, or become part of the immune system that protects you from illness. The translation from genetic language to protein language happens constantly throughout your body. Right now, as you rest in the quiet darkness, millions of ribosomes are reading genetic messages and building the proteins you'll need for tomorrow. They work through the night like dedicated craftspeople, ensuring that every process in your body has the tools it needs. Some proteins last for weeks or months, serving as sturdy structural elements in your bones and muscles. Others live for only minutes, quickly completing their tasks before being recycled into new building materials. Each protein has its own lifespan, its own purpose, its own contribution to the symphony of your existence. The remarkable thing about this cellular translation is its universality. The same genetic code that your cells read is understood by bacteria, plants, fungi, and every other living thing on Earth. A genetic sentence that means build this protein in your liver cells would mean exactly the same thing in a sunflower or a salmon. As sleep draws nearer and your cellular translators continue their quiet work, you might wonder about the rules that govern this ancient process. What determines when genes get read and when they stay silent? What invisible grammar guides the flow of genetic information through the landscape of life? Think of your favorite song. The way notes flow together in patterns that make your heart skip. The rhythm that carries you along, the pauses that make you hold your breath. Music follows invisible rules that transform simple sounds into something that moves your soul. Your genetic code works the same way. It follows grammar rules more elegant than any language ever spoken. These molecular patterns decide which genes get read and which stay silent. They determine whether a cell becomes part of your beating heart or your thinking brain. The grammar of genetics begins with punctuation marks written in DNA itself. Just like sentences need periods and paragraphs need spaces, genetic messages need signals that say start here and stop there. These molecular punctuation marks guide the cellular readers, showing them where each genetic sentence begins and ends. Some genetic punctuation marks are like capital letters at the start of important announcements. They signal to the cell's machinery that a crucial gene is about to begin. Other marks work like whispered footnotes, adding quiet instructions about how fast or slow to read the genetic text that follows. But genetic grammar goes far deeper than simple punctuation. Your DNA contains special sequences called promoters that work like stage directors. They decide which genetic actors get to perform and which must wait in the wings. When a promoter calls for action, the cellular machinery gathers around the right gene and begins the process of translation. Think of your cells as a vast orchestra. Each musician holds the same sheet music, but they don't all play at once. The conductor's baton points to different sections at different times. The violins might sing while the drums stay silent. Later, the brass section takes center stage while the strings rest quietly. Your genetic conductor is a collection of proteins called transcription factors. These molecular maestros read the cellular environment like a conductor reads the mood of the concert hall. When your body needs more energy, they direct genes involved in metabolism to play their parts. When you encounter something new, they guide learning genes to join the symphony. The grammar rules become even more fascinating when you consider that the same genetic sentence can mean different things in different cells. It's like a word that changes meaning depending on who's speaking. The gene for insulin means one thing in your pancreas cells and something completely different in your brain cells. Your liver cells read genetic instructions about processing nutrients and ignore the passages about making tears. Your eye cells focus on the chapters about seeing light while skipping over the sections about digesting food. Each cell type has learned to read only the parts of the genetic story that matter to its particular role in your body. This selective reading happens through a beautiful system of genetic switches. Like dimmer switches that control the lights in your room, these molecular switches can make genes glow brightly, fade to a whisper, or turn off completely. The switches respond to signals from inside and outside the cell, constantly adjusting the genetic lighting to match what's needed. Some genetic switches are controlled by the rhythm of your daily life. Genes that help you feel alert turn up during the day and dim as evening approaches. Others follow the slower rhythms of growth and healing, brightening during childhood and changing their patterns as you age. The grammar of genetics also includes rules about timing. Certain genes are only allowed to speak during specific moments in your life. The genes that guided your development before birth now rest quietly, their job complete. Other genes wait patiently, ready to activate only if you face injury or illness. Environmental signals add another layer to genetic grammar. The food you eat, the air you breathe, and the experiences you encounter all send chemical messages to your genes. These signals don't change the genetic text itself, but they influence how that text gets read and translated. Think of it like reading the same book in different seasons. The words don't change, but your understanding shifts based on your mood, your experiences, and what's happening around you. Your genes experience something similar, their expression colored by the molecular weather inside your cells. The most remarkable aspect of genetic grammar is its flexibility within structure. Like jazz musicians who improvise while following basic chord progressions, your genes can vary their expression while maintaining essential patterns. This flexibility allows your body to adapt to new situations while keeping your core identity intact. Your genetic grammar also includes error-checking systems more thorough than any spell checker. Special proteins patrol your DNA, looking for mistakes and fixing them before they can cause problems. These molecular editors work constantly, ensuring that the genetic story stays readable and true. As you rest here in the peaceful darkness, this genetic grammar continues its quiet work. Genes responsible for repair and renewal follow their nighttime patterns, becoming more active as your body focuses on healing and restoration. Others that drove your daytime activities slowly dim, preparing for the restorative cycles of sleep. The grammar rules that govern your genes create a language more complex than any human tongue, yet more natural than breathing. Every cell in your body speaks this molecular language fluently, following patterns that have been refined over billions of years of evolution. But even in this perfect genetic grammar, variation and change are not just possible, they're essential. Sometimes the molecular editors make tiny changes, and occasionally, these small alterations create something entirely new and beautiful. Imagine a monk in a candlelit scriptorium, carefully copying an ancient text letter by letter. His hand moves steadily across the parchment, but after hours of patient work, his attention wavers for just a moment. Instead of writing cat, he writes bat. One small change. One tiny difference that transforms the meaning completely. Your cells are like those dedicated monks, copying your genetic text billions of times throughout your life. Most of the time, they work with incredible precision. But occasionally, just occasionally, they make a small mistake. They change one genetic letter for another. They skip a word. They accidentally repeat a phrase. These cellular typos are called mutations, though that word sounds harsher than the gentle process it describes. Most mutations are quiet events, like whispers in the night. A single letter changes from A to G in one cell. The change is so small that nothing noticeable happens. Life continues exactly as before. But sometimes these tiny alterations create something remarkable. A mutation might change the color of your eyes from brown to green. Another might make your hair curlier or straighter. A different change could make you better at tasting certain flavors or give you the ability to see colors slightly differently than most people. Think of mutations as nature's way of editing her own work. Like a writer who crosses out a word and tries a new one, life occasionally experiments with small changes to see what happens. Most of these experiments are neutral, neither helping nor harming. Some cause problems, but others create beautiful improvements. The mutation process happens in several gentle ways. Sometimes, when your cells are copying DNA, the molecular machinery simply reads a letter wrong. It sees a G where there should be a C, like misreading handwriting in dim light. The error gets built into the new copy, creating a permanent change in that cell's genetic story. Other mutations occur when cosmic rays from distant stars strike your DNA. These invisible visitors from space carry just enough energy to flip a genetic letter from one form to another. You're being gently touched by the universe right now, though most of these cosmic contacts pass through you harmlessly. Chemical changes can also nudge genetic letters into new forms. The natural processes of life sometimes create molecules that interact with your DNA, encouraging certain letters to transform. It's like ink slowly changing color on an old page, shifting the meaning in subtle ways. When cells divide, they face the enormous task of copying 3 billion letters perfectly. Imagine typing a book with 3 billion characters and making fewer than one mistake per million letters. Your cellular copyists achieve this remarkable accuracy, but they're not quite perfect. The few errors they make become the raw material for genetic diversity. Your body has teams of molecular editors that patrol your DNA, looking for these small mistakes. Like careful proofreaders, they scan for errors and fix most of them before any problems arise. But they don't catch everything. Some changes slip through, becoming permanent parts of your genetic story. The timing of mutations matters tremendously. Changes that happen in your heart cells stay in your heart. Mutations in your liver cells affect only your liver. But if a mutation occurs in the special cells that create eggs or sperm, it can be passed to your children, becoming part of their genetic inheritance. Most mutations are like rearranging furniture in a room. The space looks slightly different, but it functions just as well as before. These neutral changes accumulate slowly over generations, creating the genetic differences that make each person unique. Your particular combination of tiny mutations has never existed before and never will again. Some mutations act like beneficial renovations. A change might make a protein work slightly better in cold weather, or help the immune system recognize new threats. These helpful mutations spread through populations over time, improving the species' ability to survive and thrive. Occasionally, mutations cause problems by disrupting important genetic instructions. Like a typo that changes the meaning of a crucial sentence, these changes can interfere with normal cellular processes. But even harmful mutations play a role in the grand story of life, teaching scientists about how genes work and leading to better treatments for genetic conditions. The beautiful irony is that without mutations, life would be static and unchanging. The very errors that might seem like flaws are actually the engine of evolution. Every helpful trait your species has developed, every adaptation that allows humans to thrive in different environments, began as a small genetic change in someone's cellular library. Your eye color, your height, your ability to digest milk as an adult, your resistance to certain diseases, all of these traits trace back to ancient mutations that proved beneficial. You carry the legacy of millions of small genetic experiments, most of which worked out well enough to be passed down through the generations. Even identical twins, who start with exactly the same genetic text, gradually accumulate different mutations throughout their lives. By the time they are adults, their DNA libraries contain subtle differences that make each twin genetically unique, despite their shared beginning. The mutation process continues right now as you rest. Your cells are dividing, copying, and occasionally making tiny changes to your genetic story. Most of these changes will never be noticed. They'll simply add to the quiet diversity that makes you uniquely yourself. As you breathe slowly in the peaceful darkness, consider the gentle randomness of genetic change. Each mutation is like a single note in an improvised symphony, contributing to the endless variations that make life beautiful and adaptable. But these random changes don't occur in isolation. They become part of a larger process, one where time itself becomes the patient editor, slowly shaping which genetic variations survive and flourish across the generations. Imagine an editor who never sleeps, never rushes, and never stops refining a manuscript that has been in progress for 4 billion years. This editor doesn't use red ink or strike through unwanted words. Instead, she works with the gentle persistence of wind shaping stone, water carving canyons, and time polishing rough edges into smooth beauty. This editor is natural selection, and she has been reviewing every genetic change that has ever occurred in every living thing on Earth. Her standards are simple but profound. Does this change help the organism survive? Does it improve the chances of having healthy offspring? Does it make life just a little bit better in some small way? When your ancestors developed mutations that helped them see color more clearly, the editor approved. Those changes were kept, refined, and passed down through countless generations until they became part of your genetic inheritance. When other mutations proved harmful or useless, they were quietly removed from the story, fading away like forgotten words on an old page. The editing process works with infinite patience. Unlike human editors who might review a book in weeks or months, natural selection takes generations to evaluate each genetic change. She observes how organisms with different mutations fare in the real world. Do they find food more easily? Do they avoid danger better? Do they attract mates and raise successful families? Think of it like a vast, gentle experiment that never ends. Every living thing carries slightly different genetic variations, like different drafts of the same story. Life itself becomes the testing ground where these variations prove their worth. The ones that work well get copied and shared. The ones that don't gradually disappear. Your body carries the results of millions of these editorial decisions. The shape of your teeth reflects ancient choices about diet. The length of your limbs echoes adjustments made for walking upright. The complexity of your brain shows the accumulated benefits of countless mutations that improved thinking, communication, and problem solving. The editor of evolution doesn't work alone. Environmental pressures act as her advisors, suggesting which traits might be valuable. During ice ages, mutations that helped ancestors stay warm were favored. In times of disease, genetic changes that improved immune function became precious. As climates shifted and food sources changed, different traits rose and fell in importance. Sometimes the editing process faces difficult decisions. A genetic change might help in one situation but cause problems in another. The mutation that helps your blood carry oxygen efficiently at high altitudes could make you more susceptible to certain infections. The editor weighs these trade-offs carefully, keeping changes that provide the greatest overall benefit. The beautiful thing about evolutionary editing is that it never aims for perfection. Instead, it seeks good enough solutions that work in the real world. Your eye, for example, has a blind spot where the optic nerve connects to your retina. A perfect designer might avoid this flaw, but the evolutionary editor kept this design because it works well enough for survival and reproduction. Natural selection also preserves genetic diversity, like a wise librarian maintaining multiple editions of important books. What works well in one environment might be disastrous in another. By keeping various genetic versions in the population, the editor ensures that some individuals will thrive no matter how conditions change. You can see the editor's work in the elegant solutions found throughout the natural world. The hollow bones of birds that provide strength without weight. The echolocation systems of bats that navigate in complete darkness. The cooperative behaviors of ants that build complex societies. Each represents millions of years of careful genetic editing. The editing process continues right now, even in our modern world. Bacteria develop resistance to antibiotics through rapid genetic changes that are tested and refined in real time. Viruses mutate constantly, with successful variations spreading while unsuccessful ones fade away. The editor never rests, never stops improving life's ability to adapt and survive. Your own genetic story bears the marks of countless editorial decisions made by your ancestors' experiences. The genes that help them survive famines, resist diseases, and adapt to new environments all contribute to who you are today. You carry within you a living manuscript that has been continuously refined for millions of years. But the editor's work isn't just about survival. Sexual selection, a special type of natural selection, has shaped traits that make organisms more attractive to potential mates. The peacock's brilliant tail, the songbird's complex melodies, and even aspects of human creativity and intelligence may have been refined through this gentler form of editing. The most remarkable aspect of evolutionary editing is its creativity. Working only with random mutations and environmental feedback, natural selection has written the most beautiful and complex stories in the universe. From the intricate dance of molecules in your cells to the vast ecosystems that circle the globe, everything reflects the patient work of this invisible editor. As you rest here in the quiet darkness, the editorial process continues around you and within you. New mutations arise in cells throughout your body. Most will never matter, but a few might contribute to the ongoing story of human adaptation and survival. The editor of evolution has spent billions of years refining life's manuscript, but her work is far from finished. Between the lines of every genetic sequence lie hidden instructions, waiting to be discovered and understood. Picture a symphony where the music itself is only half the story. Between the notes lie the subtle instructions that transform simple melodies into breathtaking art. The crescendo marks that tell the violins to swell. The pianissimo whispers that guide the flutes to softness. The rests that create space for beauty to breathe. Your genetic code contains a similar hidden layer of meaning. Between the genes that code for proteins lie vast stretches of DNA that were once dismissed as empty space. Scientists called them junk DNA, assuming they served no purpose. But like explorers discovering treasure in what seemed like barren land, researchers have found that these spaces pulse with quiet activity. These mysterious sequences don't spell out instructions for building proteins. Instead, they whisper instructions about timing, intensity, and coordination. They are the conductors of your cellular orchestra, determining which genetic instruments play when, and how loudly their molecular music should sound. Think of your DNA as a vast library where the books are important, but the spaces between them matter just as much. In those spaces live the librarians who decide which books get read, when they get opened, and how many people can access them at once. These genetic librarians work through molecular switches and dimmers that can turn genes up, down, or off completely. Some of these regulatory sequences act like molecular alarm clocks. They remain silent for months or years, then suddenly activate specific genes at exactly the right moment. The genetic switches that trigger puberty wait patiently through childhood before announcing that it's time for your body to change. Others control the daily rhythms that make you feel sleepy at night and alert in the morning. The spaces between your genes also contain enhancer sequences that work like volume controls on a sophisticated stereo system. An enhancer might be located thousands of genetic letters away from the gene it controls, yet it can reach across the distance to adjust how actively that gene gets read. It's like having a remote control that can fine-tune the expression of life itself. Some regulatory sequences work as silencers, gently turning down genes that might otherwise be too active. Like a parent's quiet shove that calms an excited child, these molecular silencers ensure that genetic expression stays balanced and harmonious. Without them, some genes would shout when they should whisper, disrupting the delicate symphony of cellular life. The most fascinating regulatory sequences are the ones that respond to your environment and experiences. When you exercise regularly, certain genetic switches activate genes involved in muscle building and oxygen transport. When you learn something new, other switches turn on genes that help your brain form new connections. Your experiences literally rewrite the instructions for reading your genetic story. These environmental responses happen through a beautiful system called epigenetics, which means above genetics. Imagine your DNA as a piano, and epigenetic marks as the pianist's hands. The piano keys don't change, but different touches create completely different music. Epigenetic modifications can make genes more or less accessible without altering the underlying genetic code. Some epigenetic marks act like sticky notes placed on important pages in your genetic library. They flag certain genes as "read this often" or "use only in emergencies." Others work like bookmarks, making it easier for cellular machinery to find and access specific genetic instructions when they are needed. The remarkable thing about epigenetic marks is that some of them can be passed from parents to children. If your grandmother lived through a famine, the epigenetic changes that helped her survive might still influence how your genes respond to food scarcity. It's as if life maintains a memory of past challenges, preparing future generations for similar experiences. Your regulatory sequences also control the intricate timing of development. The genetic switches that turned you from a single cell into a complex human being didn't all flip at once. Instead, they activated in precise sequences, like a molecular choreography that lasted 9 months. Some switches that were crucial before birth now remain permanently silent, their job complete. Even now, as you rest in the peaceful darkness, your regulatory sequences continue their quiet work. Genes involved in repair and restoration become more active during sleep, while others that drove your daytime activities gradually dim. Your cellular orchestra shifts from the energetic rhythms of wakefulness to the slower, more restorative melodies of night. The regulatory sequences also help your cells communicate with each other. When one cell faces stress or damage, it can send molecular signals that activate protective genes in neighboring cells. This cellular cooperation happens through networks of regulatory switches that coordinate responses across tissues and organs. Scientists are still discovering new types of regulatory sequences hidden in the spaces between genes. Some work like molecular timers, counting cell divisions and activating age-related changes. Others act as sensors, detecting chemical signals and translating them into genetic responses. Each discovery reveals new layers of meaning in what once seemed like empty genetic space. The beauty of regulatory sequences lies in their subtlety. They don't change what your genes say, but they control how that genetic story gets told. Like skilled editors who know when to emphasize a phrase or when to let words speak softly, these molecular regulators shape the expression of your genetic identity. As sleep deepens and your cellular rhythms shift to their nighttime patterns, the regulatory sequences continue their invisible work. They ensure that the right genes speak at the right times, maintaining the delicate balance that keeps you healthy and whole. But these genetic spaces hold even more secrets, and the way your DNA folds and arranges itself in three-dimensional space creates another layer of meaning that scientists are only beginning to understand. Imagine taking a 6-foot ribbon and folding it so perfectly that it fits inside a space smaller than the tip of a pin. Not crumpled randomly, but folded with the precision of a master origami artist, each bend deliberate and meaningful. This impossible-seeming feat happens inside every cell in your body, where your DNA performs the most elegant folding act in nature. Your genetic material doesn't lie flat like words on a page. Instead, it twists and loops and coils into intricate three-dimensional shapes that would make any sculptor envious. This molecular origami isn't just about saving space, though that's remarkable enough. The way your DNA folds determines which genes can be read and which must wait in silence. Think of your DNA as a vast origami crane that's still being folded. Some parts of the crane are exposed and easy to reach. These become the active genes that your cells can read and translate into proteins. Other sections are tucked deep inside the folds, hidden away like secrets written on the inside of a paper sculpture. The folding begins with the simplest twist. Your DNA double helix wraps around small protein spools called histones, like thread winding onto tiny bobbins. 8 histone proteins cluster together to form a core, and your DNA loops around each core about twice, creating structures that look like beads on a string under a microscope. But this is only the first level of folding. The beaded string then coils upon itself, forming thicker fibers that twist and loop in even more complex patterns. It's like braiding a rope, then braiding those braids together, then folding the entire structure into an intricate knot that somehow knows exactly where each strand belongs. As the folding continues, some regions of DNA come together like dancers in a molecular ballet. Genes that need to work together find each other across vast genetic distances, bending space and time at the cellular level. A gene on one chromosome might reach across the nucleus to touch a regulatory sequence on a completely different chromosome, creating temporary partnerships that control genetic expression. These three-dimensional interactions happen in special neighborhoods within your cell's nucleus. Like districts in a city where certain activities cluster together, your DNA organizes itself into functional territories. Genes involved in immune responses gather in one area. Those controlling growth and development congregate in another. The active genes tend to cluster in the center of the nucleus, while silent genes often rest quietly near the edges. The folding patterns aren't random. They're guided by special proteins that act like molecular architects, reading the cellular environment and adjusting the DNA's shape accordingly. When your cells need to activate certain genes quickly, these architects can reorganize entire sections of the genetic sculpture, bringing hidden genes into accessible positions. Different cell types fold their DNA in different ways, even though they all contain the same genetic information. A brain cell arranges its DNA to make neuron-specific genes easily accessible, while keeping liver genes tucked away in tightly folded regions. A heart cell does the opposite, exposing cardiac genes while silencing those needed for thinking or digestion. The origami changes throughout your life. During development, your cells constantly refold their DNA as they transform from generic stem cells into specialized tissues. The genes needed for early growth get folded into accessible positions, while those required for adult functions wait patiently in more compact arrangements. Even your daily activities influence how your DNA folds. Exercise can cause your muscle cells to reorganize their genetic architecture, bringing fitness-related genes to more prominent positions. Learning something new might cause your brain cells to adjust their folding patterns, making memory and cognition genes more accessible. The most beautiful aspect of genetic origami is how it creates loops and bridges across vast molecular distances. Regulatory sequences that are thousands of genetic letters apart can be brought together through clever folding, allowing them to work as a team. It's like folding a map so that two distant cities touch, creating shortcuts through three-dimensional space. Scientists have discovered that the folding patterns themselves carry information. The shape of the DNA sculpture is like a second genetic code, written not in chemical letters but in spatial relationships. Which genes touch which other genes? Which regions cluster together? Which areas remain isolated? All of these spatial questions influence how your genetic story gets expressed. Your cells can even use the folding patterns as a form of memory. When a gene has been active recently, the local DNA architecture might remain in an open configuration, making it easier to activate that gene again quickly. It's like leaving a book open to a page you might need to reference soon, rather than closing it and having to find your place again. The energy required for all this folding and refolding is considerable. Your cells must constantly invest in maintaining the proper DNA architecture, adjusting it as needed for different circumstances. Special molecular motors work like tiny engines, using cellular fuel to pull DNA strands into new positions and hold them there. As you rest here in the quiet darkness, your DNA continues its slow, graceful dance of folding and unfolding. Genes needed for repair and restoration are being moved into accessible positions, while those required for active movement and thinking are gently folded into more compact arrangements. The origami patterns in your sleeping cells create a three-dimensional landscape where genetic stories wait to be told. Some regions stand tall and exposed, ready to be read at a moment's notice. Others rest in quiet valleys, protected and patient. But this ancient art of genetic folding faces new challenges in our modern world, as scientists learn to read and even rewrite the very instructions that have been folded so carefully for millions of years. Imagine discovering that you could edit any book ever written, changing single words or entire chapters with the precision of a master calligrapher. Not just any book, but the most important manuscript in existence, the one that writes itself and has been doing so for billions of years. This is the profound gift that scientists have recently received. For the first time in human history, we can hold the editor's pen that has always belonged to evolution alone. We can read the genetic text that flows through every living thing and, with careful reverence, make gentle changes to improve the story. The tool that makes this possible has a name that sounds like the snap of scissors, CRISPR. CRISPR began as nature's own editing system, discovered quietly working inside bacteria. These microscopic organisms had developed their own way of recognizing and cutting out unwanted genetic sequences, like cellular librarians removing pages that didn't belong in their collections. Scientists learned to borrow this bacterial wisdom, adapting it to work in human cells. The beauty of CRISPR lies in its elegant simplicity. Like a word processor that can find and replace specific text, this molecular tool can locate exact sequences in your genetic code and make precise changes. It can remove a single letter that causes disease, insert helpful genetic instructions, or even rewrite entire genetic sentences. Think of CRISPR as having two main parts working together in perfect harmony. The first part acts like a bloodhound, trained to recognize one specific genetic scent among billions of possibilities. This guide molecule can find a target sequence even in the vast library of human DNA, navigating through 6 billion letters to locate the exact spot that needs attention. The second part works like a pair of molecular scissors, but far more precise than any tool humans have ever created. These scissors can cut the DNA double helix at exactly the right location, creating clean breaks that the cell's natural repair systems can easily fix. It's like having a surgeon who never misses, never hesitates, and never leaves unwanted marks. When the CRISPR scissors make their cut, something beautiful happens. Your cell's own repair machinery rushes to the scene, like skilled craftspeople responding to fix a small break in a precious artifact. Scientists can provide new genetic material to fill the gap, or simply allow the cell to heal the break in its own way. The applications of genetic editing stretch like morning light across landscapes of possibility. Researchers can now correct the single-letter changes that cause inherited diseases, transforming genetic typos that have caused suffering for generations. A child born with sickle cell disease might have their bone marrow cells edited to produce healthy red blood cells, carrying oxygen properly for the first time. Scientists approach this power with the reverence it deserves. Like archaeologists handling ancient manuscripts, they work slowly and carefully, testing each change thoroughly before moving forward. They understand that editing the human genetic story carries responsibilities that stretch far beyond any individual treatment. The editing can happen at different levels, each with its own implications. Somatic editing changes genes in specific tissues, like correcting the genetic instructions in liver cells to treat metabolic disorders. These changes help the individual but don't get passed to future generations, making them safer to explore as we learn. Germline editing represents a more profound possibility, changes that would be inherited by children and grandchildren. This power to edit the human story for future generations brings both hope and careful consideration. Should we remove genetic variants that cause early blindness? What about those that increase the risk of heart disease later in life? Beyond correcting obvious problems, genetic editing opens doors to enhancement possibilities that previous generations could only dream about. Researchers explore whether we might strengthen immune systems to resist new diseases, improve the efficiency of cellular repair mechanisms, or even enhance the brain's ability to form and maintain memories. The technology continues to evolve with breathtaking speed. New versions of genetic editing tools work with even greater precision, reducing the already small risk of unintended changes. Scientists develop methods to make temporary edits that can be reversed, like pencil marks that can be erased if needed. Agricultural applications show genetic editing's gentler face. Crops can be designed to need less water in drought-stricken regions, resist diseases without pesticides, or contain more nutrients to help address malnutrition. Rice plants engineered to produce vitamin A could prevent blindness in children around the world. The most moving applications involve rare genetic diseases that affect small numbers of people but cause immense suffering. For families carrying these genetic burdens, editing technologies offer hope where none existed before. Children with conditions that once meant shortened, difficult lives might grow up healthy and strong. As you rest here in the peaceful darkness, researchers around the world continue this careful work. In laboratories that never sleep, they practice their editing skills on cell cultures and animal models, preparing for the day when genetic therapies might heal conditions that have puzzled medicine for centuries. The editing tools grow more sophisticated each year, but the questions they raise grow deeper too. As we gain the power to rewrite the genetic stories that define us, we face choices that will shape not just individual lives, but the future of our species. Yet with such profound power to change life's fundamental code comes the weight of deciding how that power should be used, and what responsibilities we bear to future generations. Imagine standing at the edge of a vast, quiet lake at twilight. The water reflects the first stars beginning to appear in the darkening sky. Each star represents a question that humanity has never had to ask before. What does it mean to have the power to edit the very essence of life itself? The questions emerge slowly, like those distant lights. Some shine brightly with obvious answers. Should we correct genetic changes that cause children to suffer and die young? The answer feels clear and warm, like the North Star guiding travelers home. Few would argue against healing a child born with a devastating genetic disease. But other questions twinkle with more complex light. Should we enhance human intelligence, making future generations smarter than any who came before? Should we strengthen our immune systems to resist diseases that haven't even been discovered yet? Should we extend human lifespan far beyond what nature intended? These questions don't have simple answers. They require us to think deeply about what it means to be human, and whether changing our genetic story might also change something essential about who we are. Like philosophers sitting by a campfire, we must examine these possibilities with both wisdom and humility. Consider the question of genetic diversity. Throughout human history, our differences have been both our greatest strength and our greatest challenge. The genetic variations that make each person unique also ensure that our species can adapt to unexpected changes. If we begin editing our genes toward some ideal of perfection, might we accidentally narrow the beautiful spectrum of human possibility? The example of deafness illustrates this complexity. Many people see deafness as a condition to be corrected, a genetic typo that should be fixed. But within the deaf community, some see their condition differently. They have their own rich culture, their own language, their own ways of experiencing the world. Should genetic editing eliminate this diversity, or preserve it as part of human heritage? Similar questions arise around other genetic differences. Should we edit out the genes that contribute to autism, when many autistic individuals have unique perspectives and abilities that enrich our world? Should we eliminate genetic variants that increase the risk of depression, even though some forms of sensitivity and creativity might be connected to these same genetic patterns? The timing of genetic changes adds another layer of ethical complexity. Editing the genes of adults who can consent to treatment feels different from editing embryos who cannot speak for themselves. When we make changes that will be passed to future generations, we are making decisions for people who don't yet exist and cannot participate in the conversation. International boundaries create additional ethical challenges. Different cultures may have different views about which genetic changes are desirable or acceptable. Should there be global agreement about genetic editing, or should each society decide for itself? What happens when genetic enhancements become available in some countries but not others? The question of access weighs heavily on many minds. If genetic editing becomes a powerful tool for improving health and human capabilities, who will have access to these benefits? Will genetic enhancement become another way that privilege gets passed from generation to generation, creating new forms of inequality based on genetic advantages? Some worry about the possibility of genetic discrimination. If employers or insurance companies could access genetic information, might they treat people differently based on their genetic profiles? Could society develop new prejudices based on whether someone has been genetically enhanced or carries certain natural genetic variations? The concept of genetic authenticity raises profound questions about identity. If significant parts of your genetic code were designed by scientists rather than inherited from your ancestors, would you still be uniquely yourself? Would genetically enhanced humans be somehow less genuine than those with naturally occurring genes? Religious and spiritual perspectives add depth to these ethical considerations. Many faith traditions see human beings as created with intention and purpose. How do these beliefs interact with our growing power to redesign the human genetic template? Can genetic editing be seen as fulfilling a divine mandate to heal and improve, or does it represent an overreach of human ambition? The pace of genetic editing technology development sometimes feels faster than our ability to think through its implications. Like trying to steer a boat in rapidly moving water, society struggles to keep up with the speed of scientific advancement. We need time for reflection, discussion, and careful consideration of long-term consequences. Many scientists and ethicists advocate for a cautious approach, suggesting that we should proceed slowly with genetic editing, especially when it comes to heritable changes. They recommend extensive testing, broad public discussion, and careful regulatory oversight before making genetic modifications that would affect future generations. Others argue that we have moral obligations to use genetic editing when it can prevent suffering. They point to children dying from genetic diseases that could be prevented, and ask whether delaying treatments for ethical discussions might itself be unethical. The tension between caution and compassion creates difficult choices with no easy answers. The global nature of genetic editing research means that decisions made in one country can affect people everywhere. Genetic changes that enter the human gene pool in one location will eventually spread throughout our interconnected world. This reality requires international cooperation and shared responsibility for decisions about human genetic modification. As you rest here in the peaceful darkness, these questions continue to unfold like night-blooming flowers. Researchers, ethicists, policymakers, and citizens around the world engage in thoughtful discussions about how to use genetic editing wisely and responsibly. The conversations happen in laboratory meetings, university classrooms, government hearings, and family dinner tables. The ethical ink we use to write tomorrow's genetic policies will determine not just what we can do with our newfound power, but what we should do. The questions we're asking now will shape the genetic landscape that future generations inherit. But perhaps the most beautiful aspect of these ethical challenges is that they remind us of our connection to all life, and the responsibility that comes with understanding the universal language that every living thing shares. Picture a vast library where every book ever written uses the same alphabet. From the tiniest haiku to the most epic novel, from technical manuals to love letters, all sharing the same basic letters arranged in countless different ways. This is the reality of life on Earth, where every living thing speaks the same genetic language using just 4 molecular letters. The firefly blinking outside your window carries the same A, T, G, and C letters that flow through your cells. Its genes for making light use the same genetic grammar that your genes use for making proteins. The only difference lies in which genetic sentences get written and how they're arranged into the story of being a firefly rather than a human. When scientists first discovered this universal genetic language, it was like finding a Rosetta Stone that could translate between all forms of life. The genetic code that tells your cells how to build insulin is nearly identical to the code that tells a bacterium how to build the same protein. Life speaks one language, with countless dialects and accents. This shared vocabulary reveals the deep family connections that link all living things. The genes that control how your body segments during development are remarkably similar to the genes that create the body segments of a fruit fly. The molecular instructions for copying DNA work almost exactly the same way whether they're operating inside your liver or inside a mushroom. Even the most distantly related organisms share significant portions of their genetic libraries. About 30% of your genes have recognizable relatives in yeast, those tiny single-celled fungi that help bread rise and wine ferment. You and a banana plant share roughly 40% of your genetic text, both of you inheriting ancient instructions for the basic processes of cellular life. The towering redwood trees that have stood for thousands of years use genetic machinery remarkably similar to yours. Their cells copy DNA using the same molecular tools, translate genetic messages using the same basic process, and repair genetic damage with similar techniques. The main differences lie in which genetic books they choose to read and how they interpret the shared instructions. This universal genetic language allows for something beautiful and unexpected: genetic conversations between species. Bacteria have been sharing useful genes with each other for billions of years, passing genetic solutions to common problems horizontally from organism to organism. A bacterium that develops resistance to harsh chemicals can share that genetic innovation with completely unrelated bacterial species. Scientists have learned to facilitate these genetic conversations, moving useful genes from one species to another with increasing sophistication. The insulin that helps diabetics manage their blood sugar is now produced by bacteria that have been given human genetic instructions. These microscopic factories read human genetic recipes and faithfully produce human proteins, demonstrating the universality of life's molecular language. Viruses act as genetic messengers, carrying fragments of genetic information between different species and sometimes leaving traces of their genetic visits in the host's genome. About 8% of your genetic library consists of ancient viral sequences that became permanent residents in your ancestors' DNA, now quietly integrated into the human genetic story. The bacteria that live within your digestive system maintain their own genetic libraries while participating in the larger ecosystem of your body. These microscopic residents contribute genes for breaking down foods that your own cells cannot process, essentially lending you their genetic capabilities to expand your dietary options. Plants demonstrate genetic conversations on a grand scale. They've incorporated the genetic machinery of ancient bacteria into their cells as chloroplasts, the green structures that capture sunlight and convert it into chemical energy. These chloroplasts still carry their own small genetic libraries, separate from the plant's main genetic collection but essential for photosynthesis. The genetic similarities between species allow scientists to use model organisms to understand human biology. Research conducted on fruit flies, mice, and even tiny transparent worms called C. elegans provides insights into human genetic processes. The same genes that cause developmental problems in laboratory organisms often cause similar problems in humans when they malfunction. Ocean creatures reveal the ocean-spanning scope of genetic conversation. Genes that allow some bacteria to produce light have found their way into jellyfish, deep-sea fish, and countless other marine organisms. The genetic instructions for bioluminescence travel through the seas, creating living lights wherever they settle. Your own genetic library contains echoes of ancient conversations between early life forms. The genes that control your immune system bear similarities to plant defense mechanisms. The molecular motors that move materials through your cells share genetic heritage with the systems that bacteria use for swimming through liquid environments. This genetic unity extends to the most fundamental processes of life. The genes that control how cells divide, how they repair damage, and how they respond to stress are conserved across vast evolutionary distances. A gene that's essential for life in a bacterium often has a recognizable relative performing similar functions in human cells. The enzymes that copy DNA work so similarly across species that scientists can mix genetic machinery from different organisms in laboratory experiments. DNA copying proteins from heat-loving bacteria can faithfully copy human genes in test tubes, demonstrating that life's basic molecular tools are truly universal. Even the genetic code itself, the translation table that converts three-letter DNA words into protein building blocks, is nearly identical across all forms of life. With only minor variations, a genetic sentence means the same thing whether it's found in a human cell, a tree leaf, or a bacterial colony. As you rest here in the quiet darkness, billions of genetic conversations continue around and within you. The bacteria in your gut exchange genetic information with each other. Plants outside your window share chemical messages that influence genetic expression in neighboring organisms. This universal genetic language reveals that despite the incredible diversity of life on Earth, all living things are part of one vast, interconnected family. From the smallest virus to the largest whale, from ancient sequoias to newborn humans, all participate in the same molecular conversation that has been going on for billions of years. But perhaps the most remarkable aspect of this genetic unity is how it connects you personally to every other living thing, creating an intimate bond that links your story to the grand narrative of life itself. In the quiet moments before sleep, when the world grows still and your breathing deepens, take a moment to consider the most beautiful poem ever written. It wasn't composed by Shakespeare or Rumi, though it contains more emotion than any sonnet. It wasn't written on paper with ordinary ink, but inscribed in every cell of your body with the molecular letters of life itself. This poem is you. Your genetic code is a unique verse in the grand epic of existence, a combination of letters and words that has never appeared before and will never appear again. Like a snowflake that forms once in the vastness of winter and then melts back into the cycle of water, your particular genetic arrangement is a singular moment in the long story of life. The poem begins with stanzas inherited from your parents, beautiful verses that echo through your family line. Your mother's contribution carries the rhythm of her mother's heartbeat, and her mother's before that, stretching back through an unbroken chain of genetic poetry that reaches to the very beginning of life on Earth. Your father's verses add their own melody, weaving together with your mother's words to create something entirely new. But you are not simply a copy of verses written before. During the creation of your genetic poem, new lines were added through the gentle shuffle of genetic recombination. Like a poet who takes familiar words and arranges them in fresh ways, your genetic creation mixed ancestral verses into combinations that had never existed, writing new stanzas that are purely your own. Each chromosome in your cells carries a different chapter of your genetic poem. Some chapters describe the color of your eyes, the texture of your hair, the way your smile curves at the corners. Others contain more hidden verses about how your immune system recognizes threats, how efficiently your cells process energy, or how quickly your wounds heal. The remarkable thing about your genetic poem is that it contains both finished verses and rough drafts still being written. Some genes express themselves clearly and constantly, like bold headlines announcing their presence. Others remain as quiet whispers, activated only under specific circumstances or at particular times in your life. Your genetic poem carries love letters from deep in the past. The genes that shape your hands echo the hands that held your great-grandparents as children. The instructions for your heart carry rhythms that have beaten in your family line for countless generations. You are the living continuation of genetic poetry that has been composed over millions of years. Some verses in your genetic poem are shared with every human being, ancient stanzas that define what it means to be part of our species. The genes that build your brain's capacity for language, your tendency to form social bonds, your ability to look up at the stars and wonder about your place in the universe, these are verses we all carry, universal human poetry written in our common genetic heritage. Other lines in your poem are rarer, shared only with certain populations or families. Perhaps you carry verses that help your body adapt to high altitudes, inherited from ancestors who lived in mountain regions. Maybe you have stanzas that influence how you taste bitter foods, or how easily your skin produces vitamin D from sunlight. These specialized verses add unique flavor to your genetic poetry. The most personal verses in your genetic poem are those created by your own life experiences. Through epigenetic changes, your environment and choices add annotations to your genetic text, like notes written in the margins of a beloved book. The foods you eat, the places you live, the stresses you face, and the love you give and receive all contribute small edits to how your genetic poem gets read and expressed. Your genetic poem is not written in stone but flows like water, constantly being copied and shared as your cells divide and grow. Each time a cell creates a copy of your genetic text, it performs an act of molecular poetry recitation, reading each letter carefully and creating new versions that carry the same verses forward through your body. The poem contains both autobiography and prophecy. The verses about your past are written in genetic signatures that remember your ancestral journey through time and space. The chapters about your future remain partially unwritten, their expression influenced by choices you haven't yet made and experiences you haven't yet encountered. As your body prepares for sleep, different stanzas of your genetic poem become active. Genes involved in repair and restoration begin their quiet recitations, while those that drove your daytime activities gradually fade to whispers. Your cellular poets shift from the energetic rhythms of wakefulness to the slower, more contemplative verses of rest and renewal. The beauty of your genetic poem lies not just in its uniqueness, but in its connection to the vast anthology of life. Your verses rhyme with those of ancient sea creatures, echo the rhythms found in forest trees, and share refrains with microbes too small to see. You are both an individual poem and part of a larger epic that encompasses all living things. In the laboratory of your sleeping body, molecular scribes continue copying your genetic verses, ensuring that each new cell carries the complete poem forward. They work with the dedication of monks preserving sacred texts, understanding that they maintain something irreplaceable and precious. Your genetic poem will continue evolving throughout your life. New verses may be activated by future experiences, existing stanzas may be read with different emphasis, and the overall meaning of your genetic text will continue to unfold as you grow and change. You are both the poem and the poet, constantly revising and reinterpreting your own genetic story. Tomorrow, when you wake refreshed and restored, you will carry this genetic poem with you into a new day. Every breath, every heartbeat, every thought will be guided by the molecular verses that make you uniquely yourself. You are living poetry written in the most beautiful language in the universe, the language of life itself. And in the morning light, your genetic poem will continue its eternal conversation with the world, adding new experiences to its verses and contributing your unique voice to the endless symphony of existence.